Alright, hopefully that's recording. Okay, so we can run this however you want. I've got some structured stuff we can go over if that's what you'd like, or otherwise you can just ask me questions. The document that I'm showing up on the screen I just sent out as an announcement. If you want to follow along on that and take your own notes, you're more than welcome. Or if you just want to reference, know what I'm talking about, it is under today's announcement. So what do you want to do? Do you guys have specific questions you want to ask, or do you want me to go through things?
If no one else does anything, can we go to 1.2 first?
You just want to skip all of 1.1?
That's what I'm saying if anybody else wants to.
I guess. I don't really care where you go from.
Because I'm good with that, but other people are.
Alright, well how about we look through it real quick. We'll just skim it, and if there's anything you're not okay with, the biggest thing with review sessions, you gotta speak up. If you sit here and don't say anything, I'm gonna be like, cool, you came for no reason. Alright? Alright, I can describe a hydrogen bond. I can explain how two strands of DNA bind to create the double helix. The answer is no, you gotta say something. So yeah?
No. Wait. Yes, no, we're good.
H bonds.
Okay.
If the two sides are held together with hydrogen bonds in the double helix of DNA, that's why it's in this section. I can explain ways that R groups affect the structure of a protein. Yeah? I can identify the properties of water caused by hydrogen bonds. I can explain what makes water polar. I can explain what moves water against gravity up a capillary tube. No? Okay. This is the property of capillary action. So what's going on there with capillary action? That's cohesion and adhesion working together. A capillary tube is just a very, very, very small glass tube. That's all it is. Like it's itty bitty. It's only like a millimeter in diameter. But basically, capillary action is water's ability to go through a very small tube against gravity. So it uses adhesion to hydrogen bonds to the walls of whatever the tube is, be it glass or be it like the xylem inside a plant, right? And then it uses cohesion, so the water molecules are attached to each other, so that water can basically climb the sides of the tube while also grabbing and dragging onto the water molecules below, bringing it up the tube against gravity. The narrower the diameter of the tube, the farther the water can travel against gravity. Even so, we're not talking much. It can travel a foot, maybe, against gravity up a tube. Okay?
Wait,
yeah.
It's because the glass is more electronegative than the water, right?
Just because of the polarity, they're not exchanging anything.
It's more, okay.
Right, well, we don't have any exchange of electrons.
Yeah.
Josh Whitfield Bay, if you are still in the building, please come to the front office. Josh Whitfield Bay, if you're still in the building, come to the front office. Thanks.
Capillary action could not happen if the tube was composed of an entirely non-polar compound. Like, nothing would happen. But assuming the tube is made of something that has some polarity to it, it'll be able to climb it using adhesion.
Wait, sorry, can you also go over, I can explain why the R groups affect the structure of the protein.
Which one?
The third one. We already checked it off.
The R groups?
Yeah.
Gladly, but when we get to them, let's hit them.
Yeah.
So we're not here until the end of time. So R groups are the only unique part, right, within a amino acid. And as you guys saw when we went out and did it outside, you each had, like, some of them were polar, some of them were non-polar, some of them had a positive ionic charge, some had a negative ionic charge. So all of those properties impacted what tertiary structure we ended up in. You guys saw when we were out there again, like you folded up in two different ways as we rearranged the primary structure. So how the properties of the R group dictate what else it will bond to or hide from, which dictates the tertiary structure.
Okay, so we have to be able to identify if it's polar, non-polar, if it's a cation or anion, and then that is how we know
what it would do. And the thing is if I gave you like a primary structure, that's why I said you could do a plausible way it could fold up, because there's a lot of options.
Yeah.
So as long as you could just say, oh, like this positive R group would attract this negative R group, and it would make it turn like this. Like, okay, sure, that's one option. You know what I mean? Like, can you identify that there are, like, what's going to attract what's going to hide from what, that'll create a unique 3D structure.
Okay.
Good. Okay. I can explain what allows a water strider to walk on water. Do you know what those are? The weird spidery things. What are they walking on? Surface tension. Surface tension exists for humans too. The only reason we can't walk on it is because our weight is too much. We break through the surface tension because all of our weight's on one point. But if anybody's ever belly flopped in the water, you understand that if you spread your weight out across a larger area, you will absolutely feel the surface tension of water when you slap it. Okay, so we feel good about this section. Okay. All right, 1.2, elements of life. I can identify the macromolecules required by living organisms. Yes, yes, there's only 4. Proteins, lipids, carbs, nucleic acids. I can describe the function of proteins in living organisms. Shout some.
Transport.
Transport. Speed up chemical reactions.
Immune response.
Immune response. Your muscles. Hormones. Et cetera, et cetera.
If we said used as like the last bit of energy used before you died, is that okay?
No, that wouldn't count because that's not their intended purpose. That's just something, I mean, yeah, that's like a fun fact, but that's not going to show you understand the properties of proteins. Good. I can describe the function of lipids. What were the 4 groups?
Earwax.
What? Earwax, which was waxes in general. Earwax is just an example of wax. Waxes, what were the other 3?
Hormone.
Fats, hormones,
Phospholipids.
phospholipids. Good. The technical name for fat is triglyceride, or, no. Yeah, triglycerides, so be careful with just straight up calling them fats in case you see the other word. I can describe the function of carbohydrates in living organisms. What is it? Energy for humans. They just say living organisms. What other function should you include? They make it, but it's not a function of it.
Structural support.
Structural support. Cell wall in plants. Chitin in crustaceans. Think like crab shell, lobster shell. That's made out of carbohydrates. So if they say in humans, the only function is energy. But if they just say in living organisms, make sure you include structural as well. Chitin's the one that looks like it's spelled funny, C-H-I-T-I-N. People say chitin, but it's chitin. I can describe the function of nucleic acids in living organisms. What is the function? Most people just rattle them off, but what is the function of nucleic acids? What do they list as the function? Any idea?
Genetic code.
Yeah, storing of genetic information, hereditary information, passing along of hereditary information. They usually phrase it something to that effect. Yeah, most people when they see that just go DNA and RNA. Right, but what do those things do? I can describe how the R groups affect the folding of the protein. So some of these are in multiple categories. So we just went over that. So hopefully you're good there. I can identify the elements found in carbohydrates. What are they?
Carbon.
Carbon, hydrogen, oxygen. Elements found in proteins.
NCHO.
NCHO. Is that it? Sometimes, what else?
S.
Sometimes sulfur.
If you are waiting for the Allied soccer bus is here. The Allied soccer bus is out front if you are waiting for that bus.
Identify the elements found in nucleic acids.
Nitrogen, phosphorus.
CHO.
You guys read them in a weird order, but that's fine. Like I learned that proteins is CHON because it was just easier to, because I made it into like a word, and I know that that one's chunks. So that's fine. I've never heard anybody give them to me in that order, so that's fine. Good. Elements found in lipids.
CHO.
And sometimes
P.
good. I can describe how macromolecules are formed. What's the process called?
Hydrolysis.
Dehydration.
Dehydration.
Dehydration synthesis. Good. It's in the name. To synthesize means to build, to lyse means to break. I can describe how macromolecules are broken apart. Hydrolysis, to break by adding water. Hydrolysis, to break with water. Dehydration synthesis, to build by removing water. Just break the words down. You want me to go over in anything more detail? Remember, you've got to speak up. I can identify which macromolecules contain nitrogen at this point, right? Proteins and nucleic acid. If they ask you a question, like there's a practice problem on AP Classroom that asks you to identify ones with nitrogen, if you only give me one of the two, are you going to get full credit? No, not unless it only asks you for one example, right? But it's going to be the nitrogen is part of the amino group for amino acids, and it's part of the nitrogenous base, either A, G, C, T, or U in nucleic acids. I can identify which macromolecules contain phosphorus,
Nucleic acids.
nucleic acids definitely, and
Lipids.
sometimes lipids. For that one, would you have to include lipids if they asked you? No, because it's a sometimes. So if it was a question of like which macromolecules have phosphorus, you'd have to put nucleic acids, but you could not put lipids and still get full credit because not every lipid, in fact, most lipids don't have phosphorus. I can identify which macromolecules contain sulfur. What's that?
Sometimes proteins.
Sometimes proteins. What's the special fact about ones that have a lot of sulfur?
Disulfide bonds.
Disulfide bonds, which are
Covalent.
covalent, so
Stronger.
they're stronger in terms of structure, preventing denaturing. Good. Okay. Questions? How do you feel? Okay. I can describe the process of hydrolysis. What is it again? Break the word down.
Add water.
To break by
Add
adding water. What's dehydration synthesis?
To form by reducing water.
Formed by removing water.
Can you draw a picture of both?
Yes.
Like,
Yeah, I was just trying to think of what I could draw off the top of my head. I can draw an amino acid off the top of my head. So while I'm drawing this, which major biological molecule does not build and break via hydrolysis and dehydration synthesis? Say like lipids. Why not? It's true.
No.
They're also not polymers. Why do they look like a polymer? Because they're really large and they hide from water all in the same area. All right, so if this was my What's this? What is it?
amino acids.
These are amino acids. So if I wanted to do dehydration synthesis, carbon can only have 4 bonds, 1, 2, 3, 4. Nitrogen can only have 3, 1, 2, 3. The only way I'm going to get the carbon to covalent bond to the nitrogen or peptide bond with the nitrogen is if I pull the hydroxyl group from the carbon, the hydrogen from the nitrogen, they together make H2O. I now have a bond left over in each. I can put these together. That's dehydration synthesis. Hydrolysis is going to be the exact opposite. We're going to break the bonds between the carbon and the nitrogen, which means carbon's going to need one more electron to be a full valence, and so will nitrogen. We get those from water via breaking it into hydroxyl and hydrogen. So it's just reverse.
If you are in the second wave of buses, please proceed outside now. We will not be calling individual buses. This is the only announcement. If you ride the second wave of buses, you need to go outside now. Thank you.
I can identify the bonds between carbohydrate monomers. What type of bond is it overall? Okay, how do you make a polymer with carbohydrates? What process?
Dehydration.
Dehydration synthesis only makes what type of bond? Covalent. So you are fine to call this covalent. Does anybody know what the fancy word is for the specific type of covalent found in carbohydrates?
Glycosidic.
Glycosidic linkage. Put it this way, you don't have to know that. They can't take off points if you call it a covalent bond, because that's correct. But if you see the phrase glycosidic linkage, don't be scared by it. Go, oh, that's just a covalent bond. Identify the bond between proteins. That's the same way. It's just a covalent, but they call it peptide. The bond between nucleic acid monomers. Do you know that one?
That's true. No, that's a lipid.
Yeah. Phosphodiester. That's what they call that one. So phosphodiester bond. Again, what is it? Covalent. Okay, so I'm fine with you just calling them covalent bonds. It's just easier. Then you're not memorizing a bunch of unique terms.
How do you spell phosphorus?
That's a fun question. I'm going to try. There's phospho for the phosphate, and then there's the, right, on the sugar. Where is the covalent bond? This isn't a bad question. We see it in protein monomers. Carbs are pretty easy to pick out too. Where do we bond nucleotides together? So a nucleotide's a sugar, a phosphate, and a nitrogenous base. How do we put two nucleotides together?
Sugar and the phosphate group.
Sugar to phosphate. And that's where we make the covalent bond between them. Because remember, the only thing holding the two sides of the DNA together are actually hydrogen bonds, and that's just attraction-based. So the only covalent bonds found in DNA and RNA are between the sugar of one and the phosphate of the next one down. Questions on this section? All good? Okay. Properties of biological molecules. Can you describe the structural components of the monomer of a carbohydrate? It just has to be in what pattern? CH2O. One carbon and one oxygen, two hydrogens. What about of a protein? So everybody at this point should be able to recognize the central carbon, the carboxyl group, the amino group, the R group, and then that extra hydrogen at the top. Well, it doesn't have to be at the top, right? We like to turn things, but those are the four components. Structural components of the monomer of a nucleic acid, that's just understanding that it's going to be a sugar, a phosphate, and a base. I leave it general because then it's going to apply to DNA and RNA. That's why that's an easier way to remember it. Because remember, the sugar is actually different from DNA to RNA, and one of the bases is different, DNA to RNA. So if you just remember sugar, phosphate, base, it'll apply to both, and you're not pigeonholing yourself to being the monomer of just one versus the other. Okay? But could you pick it out? Remember, nucleic acids, if I show you something, and you know it's a biological molecule, and it's blatantly not a protein, not a carbon, not a lipid, it's a nucleic acid. That's how I've always done them, because they're the most complex to pick out, because they have the most outliers, like ways they can be compared to the other ones. I can describe the structural components of a phospholipid. That's knowing that it is two fatty acid tails and a phosphate group. That's going to give you that stick figure with two legs shape where the head is polar and the tails are non-polar. I can describe the difference between the bonds of starch and cellulose. Don't think you can. I'm going to just be honest. How many of you remember reading between alpha and beta glycosidic linkages in the book? Did you get it? Okay. So I don't want you to panic about this. It's not even something I want you to memorize. I just want you to listen and kind of internalize that this idea exists. I have shoved down your throat structure equals function with proteins. But it's with all biological molecules. It just doesn't come up as often in carbs. That's why you don't hear it as much. It is the same with carbs. Structure equals function. And with glucose versus starch, it is as simple as With one, the hydroxyl group is coming up into three-dimensional space. That's on a particular carbon. And on the other, the hydroxyl group is going down in three-dimensional space. That's the only difference between the two of them. Alpha is, put it this way, I wrote it down because I don't keep it memorized. Alpha is going down, beta is coming up above the ring of the carbon. But that's it. That's literally the only difference between starch, okay, and cellulose. What's funny? One we can digest and one we can't. We can digest starch, no issue. You eat it. Our system loves starch. We have enzymes that break down starch. It's delicious. Your system eats cellulose, what happens? Anybody ever eaten a lot of corn?
You can't
Next time you eat a lot of corn, pay attention the next day.
Wait, which one's alpha and which one's beta?
Alpha's down. They're opposite of what I want.
No, between
Alpha's down,
like
beta's up.
glucose and starch. Or do we not even know that?
I don't remember off the top of my head. I'll pull it up. I just
Okay.
don't remember.
But it's
Because
not
it's
that
not,
important.
no, it's not. That's why I don't have it memorized. But that small difference, we have an enzyme that fits starch perfectly, but because that small difference in three-dimensional space of where the hydroxyl group is, the same enzyme does not fit cellulose. We don't have an enzyme that fits cellulose, so you cannot break down cellulose. Can other animals? Yes. Can we? No. They actually have a hypothesis out there that that's part of what your appendix did. Had the ability to contain enzymes that broke down cellulose at one point in time when our diets were different. That's one of the hypotheses that's like out there. Okay, but what I want to hit on, I don't want you to get too stuck on the biochem of that. If you end up taking organic chemistry and bioorganic chemistry, you can really get into the nitty gritty of that as you go on in life if you want to go into that field. For now, it's just an example of harping on structure equals function and to what degree it means. Like such a small difference can completely change the way something functions inside of a biological organism. Okay? Because cellulose is purely structural, starch is purely storage in plants. Is that okay? Do you understand? You understand enough at least? I can describe the difference, I can describe the structural differences between DNA and RNA. Can you?
RNA is uracil, DNA is thymine.
Ribose versus deoxyribose, which is thymine versus,
Thymine versus uracil.
and there's one more. Single-stranded versus double-stranded. Yep, DNA is going to have two strands, RNA is one. Deoxyribose has one less hydroxyl group than ribose, and thymine versus uracil. Okay, good. I can identify an R group as hydrophobic, hydrophilic, or ionic. Do you think you can at this point after the activity?
If we need extra practice, we'll do it.
There's only 20 amino acids, so
Okay.
look them up, Google them.
Okay.
I mean, like, I can't make fake amino acids, so
Yeah, that's fine.
Google amino acids and just cover up what it says they are. Basically, if there's a whole bunch of carbon and hydrogen, it's hydrophobic. If it has a literal plus sign or minus sign somewhere in it, it's an ion. If it doesn't have either of those things, it's probably polar, but if you see nitrogen or oxygen, it's also going to be polar.
What was that one with the sulfur? That was like the weird one.
So sulfur can go either way. Sulfur can go either way. If you look at where sulfur is and its placement, and so I wouldn't put one with sulfur on a test because I think it's a little bit too complicated, but let me explain. There is an amino acid that has sulfur bonded to carbon, okay? And there's an amino acid that has sulfur bonded to hydrogen. Sulfur to hydrogen is actually considered polar. Sulfur to carbon is nonpolar because of the differences in electronegativity, so they don't act the same way. So that's why I don't include those, because you're not expected to memorize the electronegativity of sulfur.
Okay.
I can describe the effect of level of saturation on the function of lipids. How do we feel about saturation? We never talked about it in class. You just read about it.
And we go over it.
Okay. So, anytime you're dealing with a lipid that has a segment of fatty acids, like a fatty acid tail, any amount of them, you know that's carbons bonded to carbons, and then there's hydrogens filling in. Carbon can make 4 bonds, which means it can be in a chain, and some of the bonds can be double, and it can still be a chain because there's still room to attach more carbons. But if you put a double bond in between two of the carbons, it cannot hold as many hydrogens as it could before. So it is now unsaturated. It also, because double bonds are shorter, it holds the atoms closer together than a single bond does because they're sharing more electrons. It creates a kink in the fatty acid tail. So they end up all kinked and bent, whereas when it's fully saturated or has only single bonds, they are perfectly straight. Right. What that's going to do is it is going to have some impacts on the way they exist in the regular world. Saturated fatty acids that the tails are all straight are going to be able to pack more closely together like sardines. Saturated acids, unsaturated fatty acids where the tails are all kinked are not going to get as close together in a real world setting. That is why typically animal fats are naturally saturated, and that's why they're solid at room temperature. Think bacon, think lard, stuff like that. Plant-based fats are naturally unsaturated, except coconut oil and avocado, and that's why they are naturally liquid at room temperature. Biggest place this plays into is actually with your phospholipids. So picture like a plant hanging outside the last few days. We've had temperatures as low as like 50 degrees and as high as like 85 in the last 2 weeks, right? Well, what happens to, say, butter if you leave it on the counter and it gets hotter and hotter and hotter? So smelts. What happens when it gets colder? It gets more and more solid. Now picture the cell membrane of plants as the temperature outside changes. Plants can't regulate internal temperature. They don't have any way to do that like we do, right? They also can't get up and move like a snake can, right? So their problem is going to be, as it gets colder, their cell membranes become more and more rigid. As it gets hotter, they actually become more and more fluid, both of which are a problem. Rigid, they're not going to let anything in and out. Fluid, they're going to let everything in and out. So plants have to be able to regulate the fluidity of their membrane to be just right, and they do it by increasing and decreasing the amount of unsaturated and saturated phospholipids they have in the cell membrane. They need it to be more fluid. They put in a whole bunch of unsaturated ones with the kinky tails holding them further apart. They do that when it gets cold. When it gets really hot, they put in pretty much only unsaturated phospholipids so they can pack a lot closer together, and it'll help them stay solid even when it is hot out. Okay, so that's a practical application of saturation in lipids. Questions? The pentose sugar is deoxyribose versus ribose. And the difference is this has an extra hydroxyl group. So on one of the carbon positions in DNA, it's just a hydrogen. In RNA, it's a hydroxyl. And that's the difference between deoxyribose and ribose sugar. They're both just 5-carbon sugar. Nitrogenous bases, it's the thymine in DNA, is uracil in RNA. The others are all the same. And strandedness is 2 in DNA, 1 in RNA. What else? Well, here, let's do a little practice because you asked. Just okay. What do you think this would be? These are amino acids, obviously. Hydrophobic. I'm going to write nonpolar because that's easier to abbreviate. Right here? Yes. I know it's a little blurry, but can you say? Where? Do you see it? Where? Here?
Oh, just kidding.
Up here?
No, like the next one, like to the right. That was a
Here?
wrong amino acid.
Where's the R group on this one? The bottom. So what is it? What? Okay. What letter is that?
Is that an O or a C?
I don't know. Think it's a no. Well, think it's a no. Think it's a no. Think it's a no. So what's that make it? Polar. If it's a C, it's non-polar. If it's a no, it's polar. I wouldn't give you one on the test where you literally couldn't tell the difference. All right, what do we got here?
I don't know.
Wait, is that a plus sign?
Where?
In the NH?
Yes, it is.
It's an ionic.
Next one. Non-polar. Last one.
Non-polar.
Good. Okay. Questions minus whether it's a C or an A or no? Okay. Feel good about it? Cool. Structure and function of biological molecules continues. I suppose I do. Fine. Can you explain how changes in the subunits of protein may lead to changes in its structure? Not just in its structure, but what levels of structure would be impacted? If I give you a scenario, you should be able to tell me if primary, secondary, tertiary, quaternary, or all four are going to be impacted and in what ways. Okay? Yes? Think back to those practice questions. Remember, it's not always going to be disastrous. If we only are changing out one amino acid and the one we change out is of the same category, you're usually not talking that big of a change. If it's of a very different category, or if we change a whole bunch of amino acids, or we cut off a whole bunch of amino acids, then yeah, you're probably talking a pretty disastrous change. I can explain how the changes in the subunits of protein may lead to changes in function. Biggest thing, yes, structure equals function, but don't just straight up say change in structure means change in function. If they tell you what the function was in the problem, say it won't do that function anymore. Like, be specific. So for that, like catalase one, say it won't do hydrolysis of hydrogen peroxide or whatever it was. I can explain how changes in the subunits of a nucleic acid may lead to changes in the structure. What do you think of that one? It's a little bit different. What do you think of that one?
Sequence could be different.
You're going to end up with a different sequence. Good.
I was just gonna say, isn't it like the T's and the A's won't match?
Good. You've got two potential ways this could impact. If it's just a straight up change in the sequence, but everything's still matched up correctly, that's probably only going to be a change in function, right? Because that's going to be the type of mutation where it's not going to code correctly, but the DNA itself still looks fine. There are structural mutations in DNA where they stick like a T to a T or something like that. And that would be an actual structural difference because you end up with these like bumps or humps and things in the actual structure of the DNA. And then usually all the proteins and enzymes that do all the processes of your DNA don't fit correctly. A good example is that's a common error that happens in skin cancer. It's called a thymine dimer. Every place there's supposed to be a thymine, they accidentally shove two. And you end up with this little thymine bubble because it humps out all throughout your DNA. It's not great. That's why we don't aim to get skin cancer. Slather on the SPF. I can describe the directionality of a nucleic acid. What's that getting at?
5' to 3'.
5' and 3'. Where we're at right now, that's going to come up a lot more in genetics. I don't want you to panic about it right now, but you should remember at least from 9th grade that DNA is anti-parallel, that the two strands run in opposite directions. We will get way more into 5' and 3' as we go. Don't worry about it too much right now. But basically, nucleic acids do have directionality. They're not interchangeable any which way. I can describe the directionality of a protein. What does that mean?
N-terminal to C-terminal.
N-terminal to C-terminal. That it goes NCC, MCC. You can't just flip an amino acid like 180 and stick it back in. They won't bond correctly. So it has to be a carboxyl of one to an amino of the other. Good. I can identify the appropriate base pairings based on Chargraph's rules. That's just what to what and what to what.
A to T and C to G.
Yeah, we'll learn about Chargraph later. You don't need to know his name right now. It's just knowing A always goes to T, C always goes to G. I can identify the location of a growing nucleic acid strand. Don't worry about that right now. I'm not crossed out. That's going to come up more in genetics. I can describe the levels of folding found in a protein. Primary, secondary, tertiary, quaternary. What happens at each level? Those notes be took. Do you have questions about it? Yes.
You showed some examples of the
What do you mean by an example?
I don't know how they bond.
They don't bond. So basically, you know, if we go back to joining hands and we're the primary structure again, right? And you had the people that were hydrophobic. So we were saying we were only in 2D space outside, but we were saying they had to be inside the group of everybody else. They had to have somebody's hands, somebody's feet, somebody's something behind their back to be considered inside. So even though the hydrophobic ones did not need to be touching on the inside, they all had to make sure they were on the inside and have their like covalent bonds back behind them, protecting them. And the same thing occurs inside of a protein. They're not bonded. They're just all in the same area because they're forced there because it's where there's no water. Your cell membranes are the same way. Your cell membranes are in no way, shape, or form bonded together. The reason our cells hold together is because the tails of the phospholipid bilayer have to stay where they are because anywhere else they're exposed to water. So it forces them into a cell shape. Nothing's actually holding your individual cells together.
It's not tertiary and quaternary.
For hydrophobic exclusion, yes. Yep. Do you still want me to try and show you? Did that make sense? That's okay? Anything else about levels of protein structure? I can describe the structure of a carbohydrate polymer. What's that mean? You have two choices for what carbohydrates look like. Do you know?
No.
Rings.
Unbranched and branched.
Yeah, or chains. So you can have a branched chain or an unbranched chain, but rings and chains are your only options for how carbs are set up, and then yes, there are two subgroups of chains. But basically, right, you get a big linear chain that's either branched off or not, or you get your rings that are bonded together. That's your only options for carbohydrates. Good? I can describe the structure of a nucleic acid polymer. That's the nucleotides. Bonded together, sugar to phosphate down the backbone, and then if it's DNA, hydrogen bonded between the A's and T's and C's and G's down the middle. I can describe the structure of a protein polymer. We've hit on that like 45 times at this point. It's obviously a key point of this subject area. So make sure you can do that primary, secondary, tertiary, quaternary, and then the big structure equals function. I can describe the structure of a fat. Can you guys? These are harder.
Glycerol, fatty acid.
That's for what?
Triglyceride.
So for a triglyceride, it's 3 fatty acid tails attached to a glycerol molecule. For a phospholipid, it's 2 fatty acid tails, a glycerol, and a phosphate group. For steroids, you don't need to like memorize the exact structure, but you should be picturing lots of what? Do you remember what the little cards looked like when they were steroids? Huh?
Hexagons.
Hexagons, yeah, rings. They're in a ring structure, a big complicated ring structure, the steroids. And then waxes, don't even worry about it. I would never get into showing you what a wax looks like. Okay, good. What's the monomer of a fat?
Fatty acid.
There isn't one, was the right answer, but I don't know who mumbled it. Good, Jess. There isn't one. Why? They don't bond. They don't form polymers. If you can't make a polymer, you don't have a monomer. In 9th grade, I think we told you it was fatty acid to just be easy. It's not. Can you describe the structure of the phospholipid? We went over that. Can you describe the structure of a steroid? We just talked about that. That's going to be all rings, but they're still going to follow CH and O only plus phosphate. So how do you tell like a steroid apart from a big complex carbohydrate? The number of oxygens. The steroid's going to have basically no oxygen, whereas the carbohydrate has 1-to-1 carbon to oxygen in its ring structures. Questions? Okay. Can you describe how a non-polar to polar R group substitution would change a protein? Still okay there? Can you describe how a cytosine to thymine substitution changes the structure and function of DNA? So let's ask ourselves, cytosine and thymine are both what? They're both pyrimidines. They have the Y, and Y. So would cytosine and thymine be a problem? Yes. Why? So here they are. They're both pyrimidines, right? They're both pyrimidines. Here's our cytosine, here's thymine. What are these dots representing? Places where they'll hydrogen bond. Do they make the same number of hydrogen bonds? No. So where are we going to have the problem? It was supposed to be guanine, right? So we were supposed to have this, right? And now we're going to have this. So is it going to be the same width it was supposed to be? Yeah, kind of, because you changed a pyrimidine for a pyrimidine. But is it going to fit in correctly? No, you're still going to have a weird jagged edge in where it's supposed to be hydrogen bonding. So you're not necessarily going to have a backbone problem, but you're going to have a hydrogen bonding problem. What about cytosine to guanine? What if we change out this for this? You're going to have a much bigger problem because now not only will they not bond right, they also don't fit. So you're going to have a bubble on the outside of the, so you're going to have problems with the backbone and the hydrogen bonding when you do this because you flipped out and tried to put in a purine where a pyrimidine is supposed to fit. Last section, 1.6 nucleic acid. Structure of DNA we've gone over, structure of RNA we've gone over. Okay. Can you describe the structural similarities between DNA and RNA? Sugar phosphate backbone, A, C, and G are all the same. The sugar differences between DNA and RNA, one's ribose, one's deoxyribose. Ribose has the extra hydroxyl group. Describe the nitrogenous base differences. DNA has thymine, RNA has uracil. The structural differences between polymers. DNA is double-stranded, RNA is single-stranded. Structural differences in directionality. DNA has 2 strands and they're anti-parallel. RNA only has 1 strand, so it only goes in one direction. It's not an issue. They're both directional, but with RNA being just one-sided, it's not as big of a deal. Okay. What questions do you, oh, I have one I need to go over. Before I forget, I got asked this earlier. And I'll take questions from you guys. All right, I got asked to specifically review specific heat and evaporative cooling. Specific heat is a measurement you can take of a substance. It's the amount of energy you have to input to make 1 gram of a substance raise in temperature by 1 degree Celsius. Like that's the definition of specific heat. Water has a very high specific heat compared to other substances because of water's cohesive properties. The fact is, most substances don't hydrogen bond, okay?
We please have a custodian to the staff bathroom across from the admin hall. Thank you.
Dangerous. Okay, so because water forms hydrogen bonds, and yes, a hydrogen bond is individually weak, but in mass, strong, okay? You get this cohesive property between the water molecules, which means you're going to have to put in a lot of energy to get the hydrogen bonds to break before you're going to get a temperature raise in water. So water has a high specific heat. Because of water's high specific heat, we get 3 things coming from it. 1, it regulates global temperature. The fact that air changes rapidly from like when the sun's up to when it's down, and water doesn't. So our world's oceans and large lakes and things like that help to moderate global temperatures so that even though the air is changing rapidly, you don't get as big of changes. If you're sitting here yourself and saying, what about deserts that are really, really hot in the day and really, really cold at night? What's the issue with deserts? Every desert in the world is blocked by something from a major body of water. What is that thing? Mountains. There are mountains between every desert and the largest body of water near it. So water can't act as a moderator. Internally, the fact that organisms are mostly water helps with the fact that a lot of the chemical reactions we do are exothermic. It can just take in a lot of that energy and keep our internal body temperature from adjusting, even though we're releasing heat via doing quite a few of the reactions we do. And then lastly, evaporative cooling. So that's sweating in us. It's not sweating in all organisms, but in humans, it's the process of sweating. That's the fact when your body temperature is getting too high. And why is that a problem? Proteins denaturing, right? It can take that heat and it can stuff it into water and stuff it in and stuff it in and stuff it in. And remember, water can take in a lot of that heat, right, before it changes. And then that water can be put onto the surface of your body and it evaporates away. And when it evaporates away, what does it take with it? All of that energy or all of that heat. So we use it to cool ourselves off. Okay, so water's high specific heat allows evaporative cooling to be effective, regulates global temperature, regulates internal organisms' temperature. All right, now what questions do you guys have for me? Yeah, Jess? Yeah, so don't go nutty about it because here's the thing. Metallic bonding does not exist inside of you, right? It's a bonding that exists. It occasionally comes up, you know, when we're talking about other things, but life does not have metallic bonding. But metallic bonding is very, very similar to covalent, where electrons are shared. But the difference is it doesn't have to be just between two atoms. You can get many, 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 many atoms sharing like they always describe it as a sea of electrons that are kind of floating amongst all of them. And that's what gives metal a lot of its properties. Like, I don't remember all the like ductile and whatever. The fact that metal like bends and stretches and is all because it basically can act as one big unit because the electrons are just kind of flowing around with everybody. It's like a commune of electrons. Okay? You don't need to go into any more detail than that. What else?
Do we need to know cholesterol's like function in membranes?
So no, because we're going to get into it when we do cell membranes. Cholesterol is an example of a lipid. That's really its only existence in your knowledge right now. When we get into cell membranes, cholesterol is what helps maintain fluidity in animal cell membranes, so it's the appropriate amount of fluid. But for right now, just know cholesterol is an example of a lipid. And not evil, like the world teaches you. Hold on. Say it again.
We need to know the trans fat is on.
No, we don't really do too much with that. Trans fats are when you get into modifying how many hydrogens they would initially have, like adding more or subtracting more to adjust how saturated or unsaturated they are. It's not something we really get into because that's more of a thing. You don't find too many examples that occur in life. That's more humans modifying fats to our own will. So no, I wouldn't worry about trans fats.
Okay, so like the big thing we've been talking about is like R groups, like if you change the R group and it's like you're changing like the polarity of the R group or like if you ionic or whatever, that will impact the primary because you're changing it in the tertiary. Right. And possibly the quaternary. Right. But like would there ever be a change made to something where it's like for sure the secondary would be impacted?
No, because think about it. What creates secondary structure? It's hydrogen bonding from the hydrogens off of what's left of the amino group and the oxygens on what's left of the carboxyl group, meaning I don't care which amino acid you put in there, that's always the same. So you can trade out any amino acid you want and secondary structure is going to stay the same. The only way you'd mess up secondary structure is if you had a mutation that made you cut off a whole bunch of amino acids, something like that, because then the same secondary structure literally couldn't form. Other than that, we're really always dealing with primary and tertiary and then quaternary if it exists. Good. What else?
Can you go over like what kind of changes would affect the different structures?
Sure. So again, if we were a chain folded up and let's say Amanda and I are at positive and negative R groups, right? So we're attracted to each other in a bond, right? ion bond here is causing this folding shape we have right now. Well, what if the mutation changes it so Amanda's still a positive ion, but now so am I? We're going to repel. It's going to shove me way back this way. Well, now that's a completely different tertiary structure. The primary structure is going to be different too because you swapped out a different amino acid. So like it was valine, it's now isoleucine, right? And if that protein had quaternary structure, it's going to change too because these pieces are not going to fit together the same way. The only way you could have like a primary change without having a tertiary change is if we're a positive and negative ion, they swap me out for a different negative ion. This isn't really going to change. My primary is different, the sequence is different, but the actual folded up structure is probably no different. And then that be a case of it being only primary. Quaternary, it depends on if they tell you it has quaternary structure or not. Like if we don't know, you know what I mean? Not every protein has quaternary structure. What else? Yeah.
Can we just go over redox?
Redox reaction, sure. I know there's the oil rig thing. I was never taught it. I don't remember it that way. This is how I remember it. O and O. There's O and lose. There's an O and oxidize. That's how I've always remembered that. So if we have just, okay, how do we get here? What's going to produce NaCl? What's going to happen?
CO steals an electron.
Cl, so one electron from Na is going to go to chlorine, and we're going to end up with Na plus and Cl minus. So that means chlorine was what? It's more electronegative, right, but what process happened to it? Oxidation or reduction?
Reduce.
Okay, chlorine was reduced because it gained an electron. So it now has a charge of -1, and it has an oxidation state of -1 because it gained an electron, okay, meaning there's even less chance it's going to give away electrons. So its oxidation state goes lower. Sodium was oxidized. It's a +1 because it lost an electron. Now it's more protons than electrons. Okay. Its oxidation state is now going to be +1 because it is likely to lose electrons. What's that depend on? Basically, it's placement on the periodic table, right? That's going back to thinking of chemistry, realizing that sodium has 1 too many electrons to have a full valence shell, and chlorine is at 7, 1 under a full valence shell. So that's why sodium's going to lose and chlorine's going to gain. It's just based on their placement where they are on the periodic table. Do you need more than that? Is that enough? Why are they redox? A lot of what we do in biology is both. Like some things reduce, some things oxidize. So it's a redox reaction because both things occur. Okay. Honestly, like when we get to energy at all, but energy specifically in biology is just transferring electrons in a very specific manner to hand energy from place to place to place to place to place, because energy in biology is all within bonds. And you just build and break bonds and keep passing that energy to a new location. Building and breaking bonds is all about moving electrons to specific places. So it's all, redox is all we do when we do cellular respiration and photosynthesis to pass that energy back and forth places. What else? How do you feel? Yeah, you should. So quaternary structure, the example I always give is my pen. You could do whatever you want. But basically, in terms of manufacturing, this is a complete piece. This is how it's meant to come out of the factory. This is a complete piece. This is a complete piece. But like, I can't do anything with this pen piece, right? This is a useless piece independently. I can't write with it. I can't do anything, right? Neither can I with this piece. It's not until I take my 3 tertiary level proteins, tertiary level polypeptides that are completely folded up in the right shape, and put them together via hydrogen bonds, hydrophobic exclusion, ionic bonds, and potentially covalent bonds, that I now have a functional quaternary structure. So it's just cases where the individual tertiary polypeptides, while they're folded up completely, don't do anything. They don't independently have a function until they join together with other polypeptides to form one full quaternary unit that does one job together. It's only some proteins. It's just what it is. Some proteins are fully formed and functional at their tertiary structure. I don't have a pencil, but that's what I would hold up. Just like a standard old school pencil, that would be tertiary structure. The way it comes out of the factory, it's done. It's ready to use. It's in its fully formed piece. Whereas these 3 individual pieces are not done or not ready until they're put together. Did that help? Okay. What else? Do you feel better, worse, or somewhere in the middle? No response, just what I hoped for.
Better.
Okay, thank you for thumbs up. I see it now. I see it now. All right. Do you have more questions? You can go through this. There are some practice multiple choice questions at the end. They're just to make sure you know content. They're not like AP style. For AP style ones, go into the progress check on the AP Classroom. Yep, the answers are on there. And then just let me know what you still have questions about. Okay, we've got Wednesday and Thursday for periods 3 and 5, Tuesday and Wednesday, and just Wednesday for period 1. But you can still ask me questions.